This is a soapberry bug, found here on campus. These little buggers, despite their name, do not taste like soap. Now you can see, here on campus, these critters are found on two different plants, the golden rain tree and the balloon vine. Despite living on two different plants, these two populations of soapberry bugs are still considered to be the same species. Our job today is to see if there's any evidence that the soapberry bugs will differentiate into two species.
Our first line of thought, or should I say mine, was to do an observation of the two populations via a taste test. Surely, two different diets will lead to different chemical compositions in the digestive system of the insects, thus a different taste. However, due to a lack of an objective method to record taste and the lack of willingness from my lab mates to sample these exquisite delicacies, this idea was scrapped and not snagged.
Previous observations by colleagues have shown that the two populations of the soapberry bug have different average beak lengths to correspond with the average radius of the fruit of the plant to the seed pit. Balloon vine population having an average beak length of 3.98 millimeters and golden rain tree population with 3.10 millimeters. With this knowledge as context, perhaps a longer beak would require a larger size overall for the sake of proportionality or mobility of the insect. Thus, we form our hypothesis. The population of the soapberry bugs on the balloon vine will be longer in length than the bugs living on the golden rain tree, with a null hypothesis of the population of the soapberry bugs on the balloon vine will not differ in length from the bugs living on the golden rain tree.
Now, to test our hypothesis, we take to the first population of soapberry bugs, the golden rain tree population. We obtain 10 individuals in a container and bring them back to our lab station. In order to facilitate the measurement of the bugs, we have decided to subdue the bugs in an ice bath in order to take advantage of the tendency of insects to be ectothermic.
Now that these critters have been slowed down to protein popsicles, we measure the length of the bugs via calipers. We are defining the length of bugs as the distance from the tilus to the rear of the membrane, down the dorsal midline. After measuring the individual, we gently place them into a separate room temperature container for the individual to warm up. We repeat this process for the rest of the individuals for a sample size of 10. With the data collected, we repeat the whole process again with the balloon vine population. Kidnap, freeze, measure, then thaw.
Now with our data collected, we interpret it. Looking at this table, we see that the average for the balloon vine population is 9.94 millimeters, with a standard deviation of 0.785, whilst the golden rain tree population has an average of 9.55 millimeters, with a standard deviation of 1.173. At first glance, the data does seem to support the hypothesis. However, we should run this dataset through a t-test.
Using this formula and 18 degrees of freedom to account for two samples of 10, we get a t-value of approximately 0.874. This t-value is not big enough, showing that there is not sufficient evidence against the null hypothesis. In other words, the dataset does not provide conclusive evidence that the two populations will differentiate species.
This suggests there is either not enough variation or time between the two populations. Perhaps the two species are still able to flow into each other and intermingle. Therefore, further research is needed to account for potential gene flow.
Go back in. No escaping.